right?
Yes.
Okay, so did you go back and revise it? Yes, no,
No, sir. I have.
no? Okay, but you should have revised because it becomes easier to understand concepts when you have done the revisions. So, is your exams going on or something?
No, sir. They got over.
They got over, then you should have revised, both of you. What about Iram? Did you revise, Iram?
So I received the message late.
You received the message late. Okay. No worries. So let's try to let me try to brush up certain things as we go forward for you. So in the last class where we left was what all things we learned. We learned basically, we started from here. We learned about restriction endonucleases. We also learned how they work, how they make sticky ends, how they identify uh, palindromic sequences, and how can we insert and ligate our gene of interest to the vectors, right? I also told you
Yes.
towards the end how this recombinant DNA technology actually, the flow chart, basically what it is in a nutshell. Today we'll do it in detail. And then we went on to next question, that when we will be cutting our gene of interest using the restriction endonuclease from a genome, how are we going to separate the interest, our fragment of interest? So for that, we have a technique called gel electrophoresis. And I hope gel electrophoresis was clear to both of you. Now, after knowing how to, Parker is saying that my voice is cracking. Is my voice also cracking for you, Iram?
Also, no.
No. Okay. Then I think, Parker, it's your network. Though my network also sometimes get patchy. So do let me know, okay, Iram?
Sure.
Some bandwidth issue. Anyway, so in today's class, we will start with something we have actually talked about in the last class as well, which is cloning vectors. We now understand what do we mean by cloning and what are we cloning. We are cloning a particular fragment of DNA. Mostly it is a gene of interest that we want to introduce into some other organism, right? And get the protein product. Now, when we talk about cloning vectors, one thing that is very, very certain that we need as a property is it should be self-replicable, right? It should replicate within the host system independently without depending on the host's own genome. Make sense? Does that make sense, Hiro?
Yes, sir.
And we also understood in the last class that for that to happen, what should that DNA sequence have, or the vector should have?
Origin of application.
Origin of replication. Without origin of replication, we cannot make independent replication, and we cannot make copies or clone any piece of DNA inside any other organism. Now, here comes the question. First thing is, what all things can be used as cloning vectors? So one thing that we have already talked about is plasmid, right? Plasmids are extra chromosomal circular self-replicating piece of DNA in a bacterial cell. So the source of plasmids is a bacterial cell. Do you know anything else that can be used as a vector? Something that has a very high replication rate, something that can make a lot of copies of itself, which can replicate
Bacteriophage.
without. Yes, and what are bacteriophages? what the organisms they are? Like what kind of, are they bacteria,
They are
protozoa? Then what are they?
they are the virus that eats bacteria, I guess.
Yes, they are viruses. So the name bacteriophages.
Bacteria.
Is Parker saying something? Yes, Parker, you're saying something?
So she said the same
Yes,
about bacteria.
the viruses that affect the.
Yeah, yes, right. So the word bacteriophage comes from two words, the bacteria and phagy. The word phagy means eating and bacteria means bacteria. So basically they are bacteria eating viruses. Basically they kill bacteria and replicate within. So they attack bacteria. So they are viruses. Okay. Now we can either introduce plasmid inside a bacterial cell and that will replicate and make its copy, or we can introduce a bacteriophage which is engineered by us so that we remove all the genes of the bacteriophage from it and insert the genes that we need in it inside in it, and then rest everything bacteriophage will do itself. It will attack the bacterial cell, insert the genome, but this time when it will insert the genome, it will contain what we have engineered it to send. It will not contain its own lethal genes to kill bacteria, right? We just we have modified or engineered it to use it as our vector. Now both of them have origin of replications of their own, and some plasmids only have two or three or a few copies inside a cell, which means they have only very few. Their origin of replication can only make very few copies, so that's called copy number. Whereas some other plasmids and bacteriophages, they have a very high copy number. So copy number simply means how many times or how many numbers numbers it can multiply when present inside the host organism. Okay. So currently, whatever vectors we use now in the modern biotechnological age, almost all of them are engineered. It's not like we are using some native plasmid from a bacteria and which is natural without doing perturbations. We always do perturbations, right? Because we want to use it for a purpose. And for that purpose, we have to tweak a little bit here and there so that we can use it the best. Now, the question here that comes in boards is what are the characteristic features which are required in a cloning vector? So write down the question. First, write down what are the characteristic features required in a cloning vector. These are the most important feature. If any given time, if you're designing a cloning vector, you have to keep all of this in mind. So first thing, which is very obvious, and we discussed it in the beginning, is it should have its origin of replication, right?
Yes, sir.
Known as ORI, yes. So you can write down, and it's not like it just should have a origin of replication sequence. It should also support, the origin of replication should also support high copy number. Because if you have an origin of replication, you can make just one copy, okay? Because those are not very high copy number producing sequences. But the sequence, so what is this first? Write down. ORI is a sequence ORI is a sequence from where the replication process begins. From where the replication process begins. And any piece of DNA or any DNA fragment and any DNA fragment which is linked to this sequence, which is linked to this sequence, can be made to replicate within the host cell. Can be made to replicate within the host cell. So that's the definition of origin of replication sequence. But that's not all. That's not all. We also need high copy number, and this sequence is very important for controlling the copy number. Okay, so if you want to recover more copies of your DNA, then you have to clone it. You have to attach your piece of DNA in a vector that has a high copy number, that supports a high copy number. So write down. Ideally, the target DNA or the foreign DNA or DNA of interest, whatever you want to write, ideally the target DNA should be cloned. The target DNA should be cloned in a vector whose origin of replication supports high copy number. Supports high copy number. Is that clear, Parker?
Yes, sir.
Yeah. So this is the first requirement, very important. What else? What do we need in a vector apart from this? In the last class, we also talked about how are we going to make sure that our transformation has worked. Whatever we have done, it has worked. We need a selectable marker, right? We need something which we can see and we can know that transformation has happened, right? So some marker, yes or no?
Yes.
Yeah. So second important thing is a selectable marker. So what is a selectable marker? It is, again, it is a DNA sequence. Everything that is there in a plasmid or a vector is a DNA sequence, right? Origin of replication was a sequence that controls high copy number. And this
Sir?
is a sequence. Yes, Iram.
Sir,
Oh,
can
sorry,
you please
Parker. Yeah.
explain, what is a selectable marker? I think I left early last class. So I
Okay.
think I was.
Okay. So a selectable marker is apart from the origin of replication in a vector, you should also have a sequence that will produce some product when it will get translated. So it is just to confirm that your plasmid is inside the host and it is working, which means it is replicating. When it will replicate, it will make more copies. All these copies will then get transcribed and translated. And if it gets translated, then it will produce a protein if it is a gene. So a selectable marker is basically a gene that encode for any protein, which gives you a way to identify that your plasmid has reached inside with your piece of DNA. And if that has happened, then the host is transformed. The bacteria is transformed. Okay. So it is a identifying criteria to eliminate those bacteria in which our plasmid has not entered or in which the plasmid is non-functional. But in all other cells where the plasmid or the vector has entered and it is functional, can be selected, okay, accordingly. So most of the time, this gene, this selectable marker gene, is an antibiotic resistance gene. Okay, so this is a way to say that there is a colony of bacteria I am growing. Now I transform those bacteria, which simply means that I try to put my vector inside the bacteria. And if that has happened, because my vector contained a gene which provides antibiotic resistance, now these bacterial colonies will survive in the presence of antibiotic. Earlier, they were not able to survive in the presence of antibiotic. So now, after transformation, when I will plate these bacteria or when I will culture these bacteria, I will add antibiotics. And if they still survive, this is a marker for me to know that all these bacteria has been transformed with my plasmid or my vector. And if they die, it means that my vector did not enter inside, or if it enters, it got degraded, or it is not functional. Because if it was functional, it would have provided them antibiotic resistance. Does that, do you understand that, Parker? Make sense?
Yes, sir.
Yes. So write down a marker gene, sorry, a selectable marker, write down. A selectable marker helps in identifying transformants, or you can also write helps us to distinguish transformants from non-transformants. Okay. Is it clear?
Yes, sir.
And because we are talking about transformants, what do we mean by a transformant? You can also, so this word comes from the process of transformation. And I have told you above in the last class, what is the process of transformation here, right? This, it is a process by which we make the recombinant DNA enter inside the bacterial cell. Okay. And we'll see some methods how we can do it. But you can for now just write the definition that transformation is a procedure or a method. Transformation is a procedure or a method by which a piece of DNA is introduced in the host bacterium. Now, it can be a bacterial cell or it can be any other cell as well. But whenever we put any other foreign DNA in any cell, that we call that We call that method as transformation. So that DNA is transformed now. Sorry, that cell is transformed now with foreign piece of DNA. Make sense?
Yes, sir.
Perfect. So that selectable marker helps us to identify transformants and also to say that if our transformation process was a success or a failure. Now you can write down that mostly selectable markers are the genes mostly selectable markers are genes that provide resistance against antibiotics. So antibiotic resistant genes. Genes that provide resistance against antibiotics. Okay. But not all the time, okay, mostly. And with the growing need of not misusing antibiotics, scientists now are trying to use other kinds of selectable markers which are not antibiotic resistant genes. So other methods by which we can know that if transformation has happened properly or not, we'll talk about it in some time. So selectable marker is clear. What are the common antibiotics? Anyone would like to tell? What are the most common antibiotics? Which can kill bacteria and against which we can,
Penicillin.
yeah,
Penicillin.
penicillin. Yeah, penicillin, we often don't give penicillin these days. We have derivatives of it like ampicillin and kanamycin, chloramphenicol is another different antibiotic, tetracycline. So all these are antibiotics. Penicillin is also correct. And if against these, there are genes that can provide a bacteria resistance, okay, depending on how the antibiotic functions. So these are two things. What is the third thing that every cloning vector should have? Origin of replication, selectable marker, and we need a site at which restriction endonuclease cuts, right?
Yes, sir.
We call it cloning sites. If you don't have these sites or these sequences that restriction endonucleases can identify, then you won't be able to, insert anything inside the vectors, right? So third and the very important thing is cloning sites. Now, these are also called recognition sites for the restriction enzymes. And you should not have more than one recognition site for a particular enzyme in a vector. You can have multiple recognition sites against different restriction endonucleases, but not multiple recognition sites for single restriction endonuclease. Make sense? In fact, we need multiple insertions, right? I told you in the last class that we often don't just insert one thing, which is our gene of interest. We also insert what? What do we also have to insert? Selectable marker gene, right?
Yes, sir.
Yeah. So the antibiotic resistant gene is also a gene that we are inserting. So if you come to your NCRT, there is a figure that talks about very famous and commonly used cloning vector called PBR322. Can you see the image that I have inserted?
Yes, sir.
Yeah.
Yeah.
Yes. Go
So
ahead.
what's the consequence of having more than one recognition zone?
For the same uh, restriction endonuclease?
Yes.
Yeah, because the consequence is that the same restriction endonuclease will cut at two places. Because whenever you treat, so how we do the treatment, you can do sequential treatment, or if you have different sites for different restriction endonuclease, you can still control that your plasmid, after you have inserted everything, comes back to a circular form. But if you have two sites for the same restriction endonuclease, then it will cut your plasmid into two pieces the moment you treat it with a restriction endonuclease enzyme, and these two pieces will be linear, and then they won't be coming back to form a ring, right? Because they are now two linear DNAs fragments which can just float away from each other.
Yes.
Yeah. So at the end, we always need circular plasmid. So wait. Yeah, I'll just make this image slightly bigger. Yeah, can you see this image? Now this is a very common and standard cloning vector example. Now look here, the name is PBR 322. It is used for it is used as a vector for E. coli. Whenever you are doing anything in E. coli, you can use this. And you can see that it is like a full package, on it's synthesized artificially. It comes with multiple sites, like all these are as restriction endonuclease, BAM H1, SAL1, HIN3, HIN2, I already told you about, EcoR1, CLA1, PVU1, PST1. All these are restriction endonucleases, and it has one site for each somewhere here, right, in your vector. So you have the choice now. You can make the choice which one to use. Suppose you want to, insert something using BAM H1. So you can also see that what they have done already in this vector is that they have inserted, there is a sequence already. So this TET-R, this AMP-R are selectable marker genes already inserted in, so you don't have to insert it. So it comes pretty much fully loaded and ready. So this gives you protection against tetracycline. This gives the bacteria protection against tetracycline. So this is tetracycline resistant gene. And this region, this pink region, the full region, is for tetracycline resistance. And this, another pink region, is for ampicillin. So there are many ways to make this vector. How they have done it is they have put two resistant genes already. So they become your selectable marker. This is selectable marker 1 and this is selectable marker 2. They also have put, for you to make your life easy, the origin of replication, very important thing. And then this ROP is a region which codes for proteins, other proteins, so replication proteins that are needed, something extra. So it's like anything extra that this plasmid needs to be functional inside a host cell, and which the host cell cannot provide, then you have to insert all of it. It's like a package. So this is for additional proteins which are involved in replication of the plasmid. So replication proteins. And this is another restriction endonuclease site here. So it has multiple sites. Now, how can you use it is, suppose I want to insert a gene. Now I can insert my gene anywhere, wherever these sites are. If I'm using BAM H1, I will insert my gene of interest between the tetracycline resistant gene. But if I use BAM H1 or SAL1, if I use this or this, what is going to happen? if I use these two restriction endonuclease to cut my plasmid and to also cut my gene fragment, then what will happen? When I will insert it, my gene will be intact, but the tetracycline resistant gene will get fragmented. It will be inactive, right? Yes or no? Yeah. But still, what is active? Ampicillin resistant gene. So if you want to check whether your transformation has happened or not, what kind of experiment will you do? Suppose you inserted your gene of interest using BAM-H1, then you put this vector in bacteria, you cultured those bacteria and made millions of numbers of the bacterial cell. Now you want to test whether transformation has happened or not. So what experiment will you do? These kind of questions are important to make your brain work to understand the concept. What will you do?
So please could you repeat the question?
So the question is, you have used BAM-H1, restriction endonuclease, and you have inserted your gene of interest in this particular vector that you see on screen. You also did transformation, cultured the bacteria after transformation. Now you want to check whether transformation has occurred or not. What will you do? Come on, try. Please try. What will you do? First, what will happen? Let's go in process. First thing that will happen is the moment you will use BAM-H1 to do the insertion, you will inactivate tetracycline resistant gene. Okay. If your transformation has happened properly, your tetracycline resistant gene is inactive. Your ampicillin gene is still active, right? So first, to check if your vector has entered inside your bacteria, you will take some bacteria from your culture and plate it on a medium which contains both the antibiotics, ampicillin and tetracycline. If your bacteria grows in that which contains both, what does it mean?
The vector has not grown.
The vector has?
Sir, can you please repeat?
Yeah. So you have to use BAM-H1 as your restriction endonuclease to insert your gene of interest, gene X. My question is, what will happen when you will test it for transformation? How will you test it for transformation? And then I asked, if we culture those bacteria in which we have done transformation, we think we have done transformation, we think our vector has entered. Now I want to test really if it has happened or not. Then I take some of those bacteria, I try to culture it in a medium which contains both ampicillin and tetracycline antibiotics. Now what will happen if the bacteria survives in the presence of both?
So if the bacteria is able to survive in both the mediums, the vector has not gone.
The vector has? The vector has what? Come again.
So wait, I'm confused.
Yeah, this is a confusing question. That's how they're going to ask you. You have to be ready with this. Okay, let's go one by one. First scenario is you have put ampicillin, okay, but you have not put tetracycline. So tet negative and M positive. This is culture 1. Second is AMP negative, tet positive. Third is both AMP and tet positive. And fourth is
Both
both
negative.
AMP and tet negative. Okay. Now you have to tell me if your bacteria has been transformed really, okay, and you have used BAM H1, then they will, in which they will survive and in which they will not survive.
In ampicillin, they'll survive. And
I don't understand you. In
ampicillin, they'll survive.
just say
And
1, 2, 3, 4. You have 4 numbers. Just say in 1 they will survive, 2 they will not survive, something like that. Will they survive in first? If transformation has happened, if we have made a recombinant plasmid and it has really entered the bacteria, will they survive in first? Yes or no? Now you have to just say yes or no. Good evening, Rabia, for joining. Yeah, we're talking about some antibiotic resistance and how it works when we use this plasmid, which is on board. So if ampicillin is present, and if our plasmid is a recombinant, we have used BAM H1. Using BAM H1 simply means one thing, that TET resistant gene is inactive. So if TET is not present and ampicillin is present, the bacteria will survive because ampicillin resistance is still there. And this will tell us that the vector is inside and our plasmid is functional. But it still does not tell us, because we have not put tetracycline, it still does not tell us that whether the recombination has happened, whether the recombination has happened properly or not. So in second medium, when we will put where there is no ampicillin and only tetracycline, the bacteria will die. Why it will die? Because
it did not have antibiotic resistance.
against tetracycline, because tetracycline we only inactivated by putting our gene of interest here using BAM H1. So BAM H1 cleaves or cuts the gene of tetracycline resistance and put our gene in between, which makes the tetracycline resistant gene inactive. If both are present, then what will happen?
It will die.
Still it will die, because there is tetracycline and we don't have resistance against it. We only have resistance against ampicillin. And in case of fourth, where no antibiotic is there,
it will survive.
Live, but this is not giving us any information about transformation or our vector going inside, right? So we will not do this ideally, correct?
Yes, sir.
Does it make sense, everyone?
Yes.
Yeah, so this is how you have to think in order to understand what kind of questions they are going to ask you. Okay, so cloning site, all these sites that you see here, EcoR1, PVU1, PST1, PVU2, SAL1, BAMH1, all these sites are called cloning sites because basically there you can insert any gene of interest you want. Okay, so write down. Cloning sites are recognition sites. Cloning sites are recognition sites for restriction enzymes, restriction enzymes where gene of interest can be inserted in the plasmid. Where gene of interest can be inserted in a plasmid. Okay. Make sense, everyone?
Yes, sir.
Yeah. And also you can write the another question, which is asked in board multiple times. What happens if there are multiple recognition sites in a vector for the same restriction endonuclease? Okay, the answer is multiple recognition sites will lead to generation of multiple fragments of the vector. Multiple recognition sites will lead to generation of multiple fragments of the vector, which will complicate the process of recombination and gene cloning.
Which will?
Complicate the process of recombination and gene cloning. So basically, it will technically be very difficult for you to get back the vector in a circular form. Okay.
Yes, sir.
So you understood that if we have selectable markers as antibiotic resistant gene, how can we do an experiment and we can find out which are the transformants and which are the non-transformants. In this case, we will get to know when we will do the case 2 that which are our transformant cells, right? Our transformant cells will survive in case 1, but will die in case 2 and 3. Yeah, make sense? If we have used
Yeah.
BAM-H1. There is another way to select the recombinants. So there was a problem with using antibiotics, inactivation of antibiotics or antibiotic resistant gene, because of two reasons. One, we are giving antibiotic resistance to bacteria, so we are actually harboring and culturing antibiotic resistant bacteria ourselves, which we don't want to happen in the nature. So suppose you are culturing antibiotic resistant bacteria in your cell to do some biotechnological work, and by mistake you lose it to nature, or you just put it in sink, or somehow they escape or something, or it gets inside the human body. We have antibiotic resistant bacteria exposed to environment. So, and the other reason is you have to do culturing, simultaneous culturing and plating of two different antibiotics to get to know in which it survives and in which it dies, and then make a mixture of it and also check if it survives or dies. So you have to make multiple cases like we just did, right? To figure out which is our transformant colony. So there is another method which is called sorry, excuse me, insertional inactivation. So in this case, what we are doing is we are inserting our gene of interest to inactivate the antibiotic resistant genes, right? So basically, this method is insertional inactivation method. You insert something and you, because of that insertion, you inactivate something which is already present. You understood what I'm saying, everyone?
Yes, sir.
So insertional inactivation method. But what are we inactivating in the above method is an antibiotic resistant property. But what if I use an alternative selectable marker to differentiate between recombinants and non-recombinants, but not on the basis of their ability to survive in an antibiotic condition, but let's say we insert something that gives them ability to produce color chemicals in the presence of a substrate. Okay, so basically what we do is instead of antibiotic resistant gene, we use the sequence of an enzyme called beta galactosidase. So we, instead of tetracycline R gene here, we will have a beta galactosidase gene. Okay, galactosidase. Now this is not an antibiotic resistant gene. It is simply a gene for an enzyme with the same name, beta galactosidase, because you know it has A's in its name, so it's an enzyme. What does beta galactosidase do? What do you think it's do? Remember lac
So
operon?
this,
Yeah, tell me, tell me.
yeah, I was about to say lack of.
Yeah, right. So what happens here, if you have beta galactosidase gene instead of tetracycline resistant gene, and then you use a restriction endonuclease that cuts this gene somewhere in the middle, and you can insert your gene there, then this will get inactivated, right? So write down if the foreign DNA or the recombinant DNA is inserted, if the recombinant DNA is inserted within the coding sequence of this gene, beta galactosidase, if it is inserted within the coding sequence of the gene beta galactosidase, it leads to inactivation of the gene. So no gene, no enzyme, right? So this enzyme, what it does, if you have a substrate in your media, if you have a chemical, a specific kind of a chemical in the media, then if beta galactosidase is active, it will react to that chemical and produce blue color. So you will get blue colored colonies of bacteria. Okay, if the plasmid is not containing your DNA within beta galactosidase gene. So which means insertional inactivation has not happened. Beta galactosidase is intact. It is producing the enzyme, which is a protein, and that protein is reacting with the chemical that you have put in the media and giving a blue color. So if you culture the bacteria in the presence of that chemical and you get blue colored colonies, you can say that, oh, my recombination failed. I could not put my gene in beta galactosidase in my plasmid. But if your recombination has worked and your gene has been successfully inserted within the sequence of beta galactosidase, then beta galactosidase is inactive. Now, if it is inactive, there is no enzyme. If there is no enzyme, when you will culture these bacteria in the presence of that chemical, no reaction will happen. There will be no color. And then the colonies will be colorless like they normally are. And if you get normal colorless colonies, it means that those are the recombinant colonies. They successfully contain the plasmid, and they also successfully contain your gene of interest in that plasmid inserted within the beta galactosidase region. Does it make sense?
Yes.
Yes.
Yeah. It's exactly like what we did here when we used BAM-H1. We put the gene of interest within tetracycline resistance. So this gene got inactivated upon insertion of our gene of interest. Similar thing you will do here. And if you get colorless colonies, this will tell you without using any antibiotic, you don't need to plate in different cultures. You just have to do one thing, culture it, put that chemical substrate. If blue color appears over time, these colonies are blue colored dots, then your recombination has not worked. And if colonies are colorless colonies, non-colored colonies, your recombination has worked. Okay.
Yeah.
Yes.
So, why is it so weirdly named as PBR322?
Why it's named as which thing? Come again?
PBR322, the plasmids.
The plasmid. Okay, you're asking why PBR322 is called PBR322.
Yes.
Okay, so it's not all plasmids are not, I think, named in the same way, but I think why PBR is named so. So P is for plasmid because it's a plasmid group of DNA. So P stands for plasmid, and I think B and R stands for these are the discoverers initial. So it is, I'm forgetting who the discoverers were, but B is, I think, Bolivar. R, I forgot. So there are two discoverers who discovered this plasmid and figured out that it can be used. And so P is plasmid, B is first discoverer's initial, R is the second discoverer, co-discoverer's initial. So plasmid, Bolivar, something R. Just check it out on internet. And then 322 is the sequence, is the order in which they synthesized this from other plasmids, which clearly tells you that they did at least 322 experiments. Like they did it 322 times successfully to get PBR322 in the first place. So there must be something called PBR1, didn't work. PBR2 didn't work. PBR3 didn't work. PBR322, oh, this is a good plasmid, stable, it works. So that's why. RS, did you figure out R?
Yes,
Rodriguez, yes, Raymond Rodriguez. So I don't know if it's for Raymond or Rodriguez because his name was Raymond Rodriguez. Yeah. And I think it should be what is the surname? Rodriguez, no? So it should be Rodriguez.
Yes, yeah. Rodriguez.
They named it for the surname because Bolivar is also surname. I forgot his first name again. Bad with names. Yes, but that's how it's named. But there are some other plasmids which are not named in this fashion. Okay. Thank you, Iram, for that. So it's plasmid Bolivar Rodriguez 322. Okay, cool. Let's go. So insertional inactivation is clear.
Yes.
So if they give you a question on this, that in a way of through insertional inactivation method using beta galactosidase enzyme, you got blue colonies. What does this mean? What do you infer? So what do you infer? Are they transformants or non-transformants? And colorless colonies, what do you infer? First, read the question carefully. Blue colonies means non-transformants, because your piece of DNA is not inserted. Because just take the hint from the name. If insertion is successful, inactivation will happen. If no inactivation, if this gene is active, then you will get blue colonies. So you can write here gene active. Which gene? Beta gal gene. Beta galactoside is gene active. In this case, beta gal gene inactive. So non-transformants. And this is transformants. Transformants. Is it clear, everyone? So this is alternate method where you
Yes.
can use beta galactosidase as a selectable marker instead of your antibiotic resistant gene. So these three things should always be there in a good vector. Now let's talk about some of the vectors. Any one of you know what are different vectors which we can use to transfer genes into, if we want to use it, if we want to do from bacteria, if you want to do it in bacteria, we can use PBR322. And suppose we want to do transformation in plants and animal cells. How do we deliver gene into a plant cell and an animal cell? Any idea? So we go to nature and we see normally what kind of pathogens infect these cells very, very successfully. Okay. So if you want to do it in, let's say in bacteria, you use bacteriophages, because bacteriophages very successfully can infect bacteria and replicate inside them. So we can take it, modify it, use it as vector. Anything that nature has already given us, we have the power to, if we don't find any, we can synthesize from the scratch also. But that's a longer process. If you already have a template in nature, just pick it up, make some changes, and use it for your goodwill. Okay. So if you want to transform bacterial cells, we will use bacteriophage vector. If we want to transform plants, so for plants, we know that in nature there is something called a pathogen which is called Agrobacterium tumefaciens. It's a bacteria. So Agrobacterium tumefaciens. The name tells all the story. So if you go to name in biology, it will tell you the full story. Agriculture based. Bacterium, it's a bacteria. Tumefaciens means tumor inducing. Tumor, there'll be you also, tumor inducing. So it is a tumor inducing plant bacteria. Agro word talks about plant, So it is a bacteria that can induce tumor in the roots of plant. It's a pathogen. It attacks many dicot plants, and how it does it, how it functions is that it delivers or it inserts a DNA fragment from its own body, which is called tDNA. Okay, so this bacteria produces and inserts a DNA called tDNA. This tDNA is like a plasmid DNA only. And this, why it is called tDNA, the word T is for tumor. So this DNA goes into the plant cell in the root and induces tumor wherever you want to put it. Mostly bacteria lives in soil, so it induces tumor in root. But if you put this tDNA in shoot, it will induce tumor there also. So scientists figured it out and they realized, okay, let's take this tDNA and change it. Delete some portions and put some portions. So we took this tDNA, and from this whole piece, we removed the part that was inducing tumor. And in the place of that region, you can put your gene of interest. And now it becomes a cloning vector ready to use. Right? Makes sense, everyone?
Yes.
So the tumor inducing plasmid, which is the tDNA, we took it, we changed it, modified into a cloning vector. Okay, now it has no more a pathogenic role to play, but we can use it to very successfully deliver any foreign gene to a plant. Yes? No? Yeah, it's like an enemy was very successfully able to penetrate penetrate a country and was causing some problems. You figured out the enemy of of that country. You changed the enemy into something where it can no longer, go and cause any harm, but you can just use it as a postman or a messenger to send good things. You deleted the bad part. So that's what we did with this TI plasmid of Agrobacterium tumefaciens. Okay, so we call it a okay, because we just wanted to exploit the mechanism by which it delivers DNA. Similarly, so for plants, we use this. Similarly, if you want to do it in animals, tell me something that works in animals. We all know. Class, we talked about it. What has been infecting animals, including humans, for a very long time? It just did a few years ago, a few months ago, a year
Retrovirus.
ago. Not just retro, in general, viruses, right?
Yeah.
So we can use a virus. So we use adenovirus also. We use retrovirus. So adenovirus and retrovirus are different. There are rhinoviruses, which we have not used. It didn't work that way. But retroviruses are readily used. Why retroviruses are called retroviruses? Because they have RNA as their genome, right? So you can use a retrovirus, and whatever it delivers, you take that thing out, you put your desirable gene, and then allow the retrovirus to infect the animal cell. So it will do everything that a virus does. It will go bind to a cell by identifying something on the membrane. It will insert the gene or the DNA, but only the DNA this time is not the lethal DNA to produce any toxin or something. You have deleted that part and you have added, let's say, the gene of insulin or any other gene which is important that you want to deliver into the um, animal. And this is also the basis of doing gene therapy. We can use these viruses to deliver genes into a person's body where this gene was either mutated or non-functional. Make sense?
So you do what?
Gene therapy.
So we remove that gene, right? We remove the retrovirus out. So we insert a gene to remove.
So what is a virus? A virus is just a protein coat, right? With some
Yes.
mechanism to insert it. Let's say it has some mechanism by which, again, it will be a protein. And inside it will have its own genome. Now we realize that in its genome, from here to, let's say, the major part of it was lethal. Like this part was for a toxin or to make its own copies, right? So you cut that
Yes.
out and you put your gene of interest here. So basically you disarmed the pathogen, right? Now, and then send this virus to attack the cell. Now it will go attack the cell and insert this genome inside, but this time it will not have the lethal gene, right? It will not have any pathogenic gene. It will only have the genes that we have put in its DNA. So the fragment that will enter the animal cell will contain our gene of interest and not what the virus normally carries to cause infection. So no infection and gene also got delivered. Basically, you are using an enemy by modifying it as a friend now. Do you understand?
Yes. So this is also like done in the case of AIDS, HIV.
It's also done in the case of HIV. The HIV virus does the same thing. HIV virus is a retrovirus. All viruses which are retroviruses do the same thing. But we don't use HIV virus as a vector because HIV is very pathogenic. If your transformation has not worked properly, you are at a risk of, see here, if it fails, the person is not going to get HIV, right? If adenovirus vector fails, at worst, what will happen is a cold or a fever. But if HIV vector fails, the person will get HIV. You don't want to do that, right?
Can we use that as treating a patient?
Treating what patient? An HIV patient?
Yes.
So HIV is HIV is not a genetic disorder. It cannot be treated this way, where you can insert some genes in and it will work. So what is HIV? HIV already means that your body is infected. So if a body is infected by human immunodeficiency virus, that those viruses are killing T helper cells in the body, and that is how our immune system no longer functions, and we have HIV. HIV is AIDS, which is acquired immunodeficiency syndrome. Once a person has AIDS, suppose now you take HIV virus or any virus and tweak it, what are you going to send inside? You are only going to send a DNA inside. What DNA should I send inside that it will kill all the virus? We don't have anything like that, right? We can send a gene inside to make its product, or to replace, or not to replace, but to what do you say? Take the place of a non-functional gene. We can do these things. But to kill a army of viruses which has already been inside the body, by this method we can't do that. Make sense?
Yeah. Yes.
Yeah. So we use retroviruses here. We also use adenoviruses to do biotechnology in animal cells. Now, the next big question is, everything said is good. It looks in which you can make a host cell take up the plasmid. Why should a host cell take the plasmid in? Suppose you are not using these natural methods, which are natural, so host cell cannot fight against it. It can, but in a long run, in an evolutionary term. So currently, if you change Agrobacterium tumefaciens and make it into a plasmid, and you send that plasmid into a plant, it will enter the plant because plant are being infected by Agrobacterium tumefaciens. After millions of years, maybe plant will develop a mechanism to, be resistant against it. Then bacteria will also make a counter arrangement to infect it. This is like a Tom and Jerry game, the mouse and the cat. One evolves, the other evolves, because they have to co-evolve. Viruses have been affecting us from millions of years, and we have been fighting them back from millions of years, but still they affect us, right? So we find a way, they find a counter way. We find a way, they find a counter way. Similarly, it will work. But suppose you want to send plasmids. Plasmids are a bacterial product. We isolated them from bacteria, or we synthesize them artificially. Why will a plant and an animal, or even a bacterial cell, take up a plasmid? Plasmid in itself is not an organism. It does not know how to go inside on its own unless the host cell takes it. And the host cell will not take it by its own will, because the host cell does not recognize it. Okay, if I put plasmid in a solution where animal cells are being cultured, do you think animal cells will allow plasmid to come inside? No. Why will they? For them, it's a foreign particle. They don't know if they're harmful. So cell has no receptors that will bind to this plasmid, and there will be a crosstalk that, see, I am here for your benefit, so let me in. Cells will just simply not allow that to happen. So we have just one way. Suppose you have you want someone to tell you the truth, and your pleading is not working. What are you going to do? You want the work to be done. Yeah, tell me. Threaten, right? Yeah, threatening, like verbal threaten will not work on cells. Cell can't listen. So what you're going to do is you're going to make them competent. Making them competent simply means torturing them chemically. Okay. And this exactly is the process of competent, making a cell competent. So let's write it. We have to make the host cell competent enough. Competent means which is able to take the recombinant DNA so that we can transform that cell. So we make competent cells, competent host cells. Now there are multiple ways to do it. Because your DNA, and why can't DNA just enter inside the cell or any cell? Let's come to DNA's chemistry. Why it is very difficult for a DNA molecule to just pass through the cell membrane without the cell knowing? But DNA is very small, right? Can it not just pass through the cell membrane? Let's say there is a small hole somewhere. There will be some hole, some protein channel. Can't it just move inside? It can't, because DNA is hydrophilic molecule. DNA is soluble in water, right? And what is cell membrane made up of?
Phosphobilipid.
Sorry, come again.
Phosphobilipid.
Lipid molecules, and lipids have a hydrophobic moiety towards outside, right? Sorry, towards inside. So if you see a cell membrane arrangement, chemically, it's like this. You have lipid heads on both sides. Let's say this part, the DNA can interact with, okay. But what is inside? What is this double layer? The thick region. This is all hydrophobic tail, right? It does not want to interact with water. So this part is hydrophobic, and DNA is hydrophilic. So that's why a DNA cannot just pass through the gap and go in, because these hydrophobic molecules will not allow it to interact. Hydrophobic molecules do not interact with hydrophilic molecules, right? They repel it. Make sense?
Yes.
Yeah, so that's why a DNA is hydrophilic. It cannot pass through the cell membrane. So we have to find some other ways. So the other way is, yeah, go ahead.
So in 11th fluid mosaic model, we had also peripheral proteins and certain channel proteins that can allow the passage of molecules.
That's very good. Yes, right. So they are called transmembrane proteins or peripheral membrane proteins. So if a protein is passing all the way from the top to the bottom like this, it's a transmembrane. If it is like only in half, it is membrane bound or membrane embedded. You're right that there are transmembrane proteins that allow movement of things, but how they allow the movement of things is by specific binding. They have pores or grooves in which things can bind, and there is no transmembrane protein that can allow binding of a DNA fragment as a whole. So they can allow movement of some other smaller proteins, not proteins actually, some cations, anions, water, and other molecules, but they cannot allow DNA to go inside. So for DNA to go all the way through the membrane, you need something that interacts with DNA and then flips inside, or let the DNA move inside through it. So that doesn't normally happen if DNA from outside can so easily go inside, what is the risk? If you have a gate for DNA to just move in or out, what is the problem? We also have DNA in the cell. You would not like your DNA to just wander out just like that. Yeah. So what you are protecting inside, how you are protecting it? By not letting it go outside so easily. So if you allow DNA to come in so easily, DNA can also go out so easily. That you don't want. See, that's why whenever glucose can go inside the cell, right, by facilitated diffusion.
Yes.
So whenever glucose go inside the cell, what happens? What is the first thing we do to glucose? We bring a phosphate. No, before breaking up, we bring a phosphate, the first step of glycolysis. We don't want glucose to stay as glucose anymore, because if glucose can so easily come in, and if it stays as glucose, it will also go back. You lose it. So the moment glucose come in, you put a phosphate, throw it at the face of glucose and make it glucose 6-phosphate. Done. Now it's no longer glucose. It's glucose 6-phosphate, cannot go out, because the channel only allows glucose to cross. Make sense?
Yes.
That's how cells work. Beautiful, right? Good mechanisms.
Is it the same with RNA?
It's the same with yes, nucleic acids mostly, both DNA and RNA. Yeah. So that's why the viruses that infect cells, they have a different machinery of proteins, which are actually called drilling proteins. So what they do, they drill through, they physically drill. It's like a driller. They physically drill through the membrane by interacting with some proteins, and channel to inject their DNA inside the cells they are infecting. Okay.
Yes.
Yeah, they're called hole-in proteins. Hole-in means they're drilling a hole, something like that. Or pore-in, they are making a pore. Yeah, okay, cool. So to make competent cells, we have methods. The most common is you use divalent cation, concentrated divalent cations like calcium. Calcium is a divalent cation. So when you treat a cell with divalent cations, what it does, divalent cation is a big cation, right? With valency 2. So they make the small pores which are there in the bacterial cell wall bigger. Okay, because calcium just increases the efficiency with which these DNA molecules can just cross through, because now the cell wall is covered with divalent cation which are positive. Since DNA is negative, it can get attracted towards calcium, and since calcium is going inside the membrane and making the pores bigger, it's like if you cannot go through a bush, you ask your bulky friend to use force and try to go through the bush, and you are latched to your friend and you just go with your friend. Do you understand?
Yes.
It's a forceful method by treating by divalent cations, and you increase the chance of the DNA entering. Of course, this will not keep the cell in a competent state forever. It just happen not instantaneously, but for a very small time, like 30 seconds to 45 seconds. That's all. If you keep it in, if you treat it with for longer times, again the pores will close, or the DNA which has entered inside will start coming out due to the same reason. If calcium can just freely go inside, calcium can also come outside, and with it the DNA can also come outside. So what we do is, this is called a flash method. You treat with divalent cations, and with that you add your DNA. The moment it goes in, then you stop after some time the treatment. So you just want it to go in and then all the pores to close back. So even if few DNA molecules enter inside, we know that these DNA molecules have origin of replication because they are plasmids or vectors. They will make copies, so not a problem. Okay. So you understood why we make competent cells? Yeah.
Yes.
So write down what is a competent cells. Write down. Since DNA cannot pass through the cell membrane, since DNA cannot pass through the cell membrane, to force the bacterial cell, to take up the plasmid, to force the bacterial cell to take up the plasmid, it is treated with specific concentration of divalent cation, like calcium. You can also use magnesium, or you can also use a mixture of both, or you can also use any of the other element. I think what people have also used is in the periodic table, what comes in the divalent group: magnesium, calcium. You can use that also. I'm forgetting the name. Yeah, but sorry. Chromium, I don't know if people have used, but it starts with R something or Z. Anyways, but it has to be a divalent cation, and the bigger the better. But sometimes very big ones don't enter themselves because you can also not make such a big hole in the membrane. So mostly what is used is calcium and magnesium, or mixture of both. Okay, when I make it in the cell, I use mixture of both. 200 millimolar calcium and 200 millimolar magnesium together, and it works very well. So it has to be treated with specific concentration of divalent cation like calcium, which increase the efficiency of DNA entry, which increase the efficiency of DNA entry through the cell wall of the bacteria, through the cell wall of the bacteria. Cell wall and cell membrane both of the bacteria. Okay.
Yes.
And as I said, it has to be very fast. So how we do it is, we do it through heat shock. Now, what is a heat shock? It is simply you have your divalent cation and you have your DNA ready. Okay. And then you keep all your cells chilled. You keep it on ice all the time. So they are cold, 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 cold. And just for 15 to 45 seconds, anywhere between 15, 30 or 45, these are mostly the 3 times people use. I have seen that it works the best at around 40 seconds. So for just brief period of time, you take these cells out from ice, so they must be chilled, and then put it in 42 degrees. Now 42 degrees, not very hot to kill the bacterial cell, but it is hot enough to give a shock, right? You are coming from 0 degree. So ice is 0 degrees, right? Near 0. Okay. If not sub-zero, it is almost near 0. So from 0, so it's like keeping your finger in ice. It's chilled. You can't sense anything. And then for a very brief period, you keep it in 42 degree water. You will get a temperature shock, right? That's called heat shock. It will not harm you because 42 degrees is not very hot to harm, but it is hot enough to make all the pores whenever of the bacteria for very small amount of time inflate. And this is the time we were waiting for. The moment these pores inflate, we have calcium that will take along DNA with it and enter inside the cell. Make sense?
Yes.
That's called a heat shock method. And then the moment you think it's done, after 40-45 seconds, put back on ice. Why that is important? Because if you delay in putting back on ice, if you keep it in 42 and you think the more I keep it on 42, the better it is. No, that's not the case. Because if you keep it on 42, the pores will stay open for longer. And in the same manner, calcium entered inside with the DNA. It will start coming out because, you understand, right?
Yes.
The only way you are determining directionality to this process is by doing it in a flash. That's why it's called heat shock or flash shock. Put it on ice, keep it cool, put it on 42, and then put it back on ice so that everything again shuts back and all the DNA, all the recombinant plasmid DNA that has entered inside gets stuck inside. So this is one way. This is called heat shock method. Okay, so I think we are out of time, sadly. So one is heat shock. Read about heat shock plus divalent cation from book. In tomorrow's class, the next class will be the last one because we have less to cover now. All the concepts are mostly covered. We have just one concept, PCR, which I was thinking to finish today, but couldn't do that. Anyways, so we'll finish principles and come to applications in the next class for sure. But I will also teach you other methods like micro injections. If you cannot do heat shock, because heat shock works with bacteria, it doesn't work with plant and animal cells mostly. So micro injection method, or there is a method called gene gun. These two methods we'll do in next class, and then we'll come to PCR amplification.